Hi, this is the AP Bio lecture on nucleic acids, and your essential question is, what are nucleic acids composed of, and what do they do in the cell? You should be able to answer this question using these vocabulary by the end of this lecture. So first, we're going to briefly talk about the types and functions of nucleic acids, and then we'll get into the characteristics of nucleic acids, and we'll mainly talk about the structure of the nucleotide, which is its monomer. So you guys should know, of course, that nucleic acids store, transmit, and express hereditary information. And there are two main types of nucleic acids. There's DNA and there's RNA. And you might remember from ninth grade biology that there are three types of RNA: mRNA, rRNA, and tRNA. So let's take a look at those in action. This is a eukaryotic cell. You can tell because here's the nucleus. If it was a prokaryote, there would be no nucleus. But whether it's eukaryotic or prokaryotic, all cells have DNA. So this is one type of nucleic acid. And by the way, the word DNA stands for deoxyribonucleic acid. And the word RNA stands for ribonucleic acid. And so DNA is a nucleic acid that's double-stranded. And inside the nucleus, it's used to make mRNA, which is one of the types of RNA, which then leaves the nucleus. And in the cytoplasm, it joins up with yet another type of RNA called rRNA. And rRNA stands for ribosomal RNA, which folds up to make the ribosome. tRNA is not pictured, but tRNA molecules is yet another type of nucleic acid that would come into the ribosome, bringing in amino acids and using information coded in the mRNA. So basically looking at the sequence of nucleotides in the mRNA to then put together the amino acids in the right order. Those amino acids form what we call a polypeptide, which then folds up into a functional protein. But the nucleic acids that are in this model here are the DNA and the RNA. Here's one RNA, here's another RNA, and then a third one not pictured would be tRNA. What are nucleic acids made of? So nucleic acids are made of monomers called nucleotides. And when you put nucleotides together into a chain, they're called polynucleotides. So here are our two polynucleotides, our double-stranded DNA and our single-stranded RNA. And each of these little rungs in this ladder or in this comb is a nucleotide. So here's a different version of that schematic. Here you can see one single-stranded polynucleotide, and we've got 1, 2, 3, 4 nucleotides in this polynucleotide pictured here. If we zoom in on just one nucleotide or one monomer, here's what you would see. You would see a pentose sugar, pentose meaning 5-sided sugar. And you'll notice that this pentose sugar has the carbons numbered. So remember, the carbons are not like actually explicitly labeled, but anytime you see a vertex in this type of drawing, there's a carbon there. So we have 5 carbons in this pentose sugar. And on the 5th carbon, right here, we've got a phosphate group attached. Notice that it's negatively charged. And then on the 1st carbon, we have a nitrogen base attached. This is also called a nitrogenous base. This is also called just a base. So the sugar and the nitrogen base together are called the nucleoside. That's a word that you might see every now and then, but we're typically going to use the word nucleotide, which is what you get when you take these two pieces and add a phosphate to them. Now, remember that there are two types of nucleic acids. There's DNA and there's RNA. And the difference between them, there's a few differences, but one of the main differences between them is the pentose sugar in DNA is missing an oxygen right there that it does have in RNA. So you'll notice in RNA, that second carbon has a hydroxyl, but in DNA, that second carbon does not have a hydroxyl. It's missing an oxygen, and for that reason, it's called a deoxyribose. D as in missing, oxy as in oxygen. Okay, so remember that in addition to the pentose sugar and the phosphate, you have the nitrogenous base, and there are 5 types of nitrogenous bases. Cytosine, thymine, and uracil are all single ring nitrogenous bases. They're called nitrogenous because they have nitrogens in them, as you can see. And these 3 in particular are known as the pyrimidines. Over here, you have adenine and guanine, so those are our last 2 types of nitrogenous bases. And notice they have a double ring fused structure. So this is a double ring nitrogenous base. And these 2 are known as the purines. So remember that. Purines are double ring nitrogenous bases. Pyrimidines are single ring nitrogenous bases. One way that you might remember that is pyrimidines have that Y in them, and cytosine and thymine both have a Y in their names. And so cytosine and thymine are the pyrimidines that you see in DNA. But if you're looking at RNA, you wouldn't have thymine, you would have uracil instead. And you should remember from 9th grade that pyrimidines can only bond with purines, hydrogen bond, to form that DNA ladder. So T can only bond with A, G can only bond with C. There's a reason for that you're going to learn about when we get to our gene expression unit later in the year, and it has everything to do with what can go wrong during a process called DNA replication. So you'll learn more about that later. Here are your key takeaways. Nucleic acids, also known as polynucleotides, store and transmit genetic information to direct protein synthesis. Monomers are called nucleotides, which bond together to make a sugar-phosphate backbone with nitrogenous bases ordered in a specific sequence. And that specific sequence of nucleotides is going to be what causes the amino acids to get put together in the right order at the ribosome. Remember that for nucleotides, there are 2 types of nitrogenous bases that you'll see. Some are called purines, and these are bases with double rings, A and G, adenine and guanine. The others are pyrimidines, and there are bases with single rings, so cytosine and thymine in DNA, and cytosine and uracil in RNA. One thing that I kind of glossed over is this here. You'll notice that the sequence of nitrogenous bases is different, but every nucleotide has a phosphate and a sugar, which then links to the next phosphate and the next sugar, which then is bonded to the next phosphate and the next sugar. We call this the sugar-phosphate backbone. The DNA sugar-phosphate backbone is shown in blue, and you can see there are 2 of them. The RNA sugar-phosphate backbone here is shown in pink. Okay, if you guys would please go back to the essential question, see if you can answer this question using this vocabulary. And if you can, that means that you have mastered the basics of nucleic acids, which will prepare you to dive more deeply into it when we get to our gene expression unit.